All right. Hey, you made it back to the last segment. So now we're going to look at other ways to deal with catabolism. Okay. Now, there are different ways to catabolize or break down glucose to make ATP. The traditional route that we've looked at is cellular respiration. You go through glycolysis, citric acid cycle, and then oxidative phosphorylation. That happens when you have lots of oxygen. But are there circumstances where cells don't have sufficient oxygen to continue to power things? Well, yeah. So then we're going to look at some other ways to make ATP. So we'll actually start by jumping back here to, let's use this image. If we look with electron transport, again, you've taken your food, broken down your food, stripped away the electrons, and put those in NADH and FADH2. Those electrons and their associated protons go to the electron transport chain. The protons flow down their chemiosmotic gradient. They spin ATP synthase, and it brings together ADP and phosphate to make ATP. In this process, the protons and electrons from our food join with oxygen to form water. But what happens if you don't have enough oxygen? If you start to run low on oxygen, will this process continue? No. And then that shuts down basically the activities inside the mitochondria. You back up the electron transport chain, you stop making all that oxygen, that backs up or shuts down the Krebs cycle or citric acid cycle. And so that puts a world of hurt on somebody. That's why we have to keep breathing oxygen throughout our lives. You take 15, 20 breaths a minute, and you do that because you need oxygen at the end of the electron transport chain. But can we survive short periods of time without oxygen? Well, yeah. So now let's look at what happens when the electron transport chain gets shut down. All right. There are other sets of events like lactic acid and alcohol fermentation. Sorry, my dogs are startled by something horrible, I'm sure. So let's look at the idea of lactic acid fermentation. This is what happens to us during high intensity activity. So if you're sprinting, lifting heavy weights, doing something that engages a lot of muscles. So in the case of high intensity activity, you're going to go into oxygen debt. You're not going to have enough oxygen to send to your mitochondria to power the electron transport chain. So electron transport shuts down, citric acid shuts down. What do we have left? Glycolysis. If you remember that old process, it doesn't require molecular oxygen, right? Glycolysis has been around since before there was a lot of oxygen in the atmosphere. So with glycolysis, we can still make a little bit of ATP and keep things going, keeps the lights on, right? But we run into a bit of an issue. In glycolysis, we break down glucose into pyruvate, right? But we can't send the pyruvate anywhere, right? If you've shut down electron transport and the Krebs cycle, what are you going to do with that pyruvate? You can't let it accumulate, correct? Because if you let molecules or products accumulate, you're going to shut down pathways. So you got to get rid of the pyruvate. Additionally, we need enough NAD plus to accept those electrons that get stripped away from glucose during glycolysis. So we got to get rid of pyruvate, and we have to regenerate NAD plus so you can keep stripping away protons and electrons from glucose and keep glycolysis running. So the way our bodies have evolved to handle that is lactic acid fermentation. So what our bodies do during periods of high intensity activity is we take this 3-carbon pyruvate and convert it to 3-carbon lactate or lactic acid. This is pyruvate or pyruvic acid. We convert it to lactate or lactic acid, and then we send that to our livers. Our livers will take the lactate, and then when oxygen levels go back up in the body, we convert the lactate back to pyruvate, and everything's peachy. But this is important because when you convert pyruvate to lactate, what that does is it takes these protons and electrons away from NADH and it regenerates NAD plus. If you get rid of pyruvate, if you regenerate NAD plus, then you keep glycolysis running. And again, you don't get a lot. You only get 2 ATP per molecule of glucose. So you don't get a lot of ATP. But what happens is, since this is only a couple of steps, you make it really quickly. So you can make a little bit of ATP really quickly. Now notice it's a 3-carbon pyruvate into 3-carbon lactate. You don't strip away any carbon, so it doesn't make any CO2. So again, you only get a couple of ATP per glucose, but you make it really quickly. And we can do this for a short period of time, about 45 or so seconds. Okay. So we can run lactic acid fermentation for 45 seconds to a minute. So that's why, super high intensity activities, we can only do them for short amounts of time. Now, other organisms like yeast, think of Saccharomyces cerevisiae. Those are the yeast that are used to like brew beer or used in baking. They don't have the same enzymes that we do. They do a different type of fermentation. So again, they do glycolysis too. And normally glucose gets converted to pyruvate, goes to the mitochondria, and you make lots of ATP. But in the case of inadequate oxygen for organisms that perform alcohol fermentation, they take pyruvate, convert it to this 2-carbon acetaldehyde, and then that ends up being converted to ethanol, which is a 2-carbon molecule. So you start with 3-carbon pyruvate, and then you wind up with 2-carbon ethanol. And so that is a CO2 producer. So that's why like bread will rise during its proofing, is because these organisms will perform or create CO2, and that'll help with that rise or bubble creation. Let's see. Again, this is something where it's only a few steps, so you don't make as much ATP, but you can make it pretty quickly. Another issue here is that the accumulation of ethanol can disrupt metabolic pathways, so it ends up killing those yeasts or those microbes that perform the fermentation. That's also why we can use like 70% alcohol and use it in hand sanitizers. Okay. So again, it's just a different way of dealing with a lack of oxygen. Another molecule or a set of reactions that can occur is phosphorylation with creatine phosphate. Now, you may have heard of creatine phosphate as referred to as just creatine. Sometimes people will use creatine for like high intensity activities like lifting weights or playing sports that have really short duration, high intensity activities. And the reason for that creatine phosphate is that the creatine performs what's called substrate level phosphorylation. With the substrate level phosphorylation, one molecule literally just hands its phosphate off to another molecule. Okay. So in the case of creatine phosphate, what it does is it just hands off its phosphate to ADP, and that generates ATP. So this is the quickest way to make ATP, is phosphorylation with creatine phosphate, because it's a one-step reaction where you just hand off that phosphate to ADP to create ATP. So this can work really quickly. It's just for short durations of time, only 5 or 10 seconds. So people take creatine phosphate because it's a way to quickly regenerate ATP for muscles to continue to contract. So, what do you get out of it? If you're lifting weights, you might be able to get like one more rep out of a set. Or you may be able to do a high intensity activity for, just a few more seconds. But the idea is, you can phosphorylate ATP and keep muscles contracting through the substrate level phosphorylation, where creatine phosphate just literally hands off its phosphate to ATP, and you can continue to power reactions. Okay. So between creatine phosphate and lactic acid fermentation, we can perform really high intensity activities for about a minute or so. So you can do like peak effort, maximum effort for about a minute before you have to, stop the activity and continue to breathe and try and repay the oxygen debt. That's why running like a 400-meter dash is so challenging. People will sprint for 400 meters and it runs, in the ballpark of 60 seconds. So people are sort of maxing out what a person can do in terms of high intensity activity when they perform an activity like that. Let's see. Now, this is sort of an interesting way to look at things. We have reviewed sort of the traditional pathway for the production of ATP through cellular respiration. You start off with glucose, goes through glycolysis, then you have the link reaction, make acetyl-CoA, and then that goes inside the mitochondrium for citric acid, and then oxidative phosphorylation with the electron transport chain. But just remember, acetyl-CoA is just a 2-carbon molecule, right? Well, think of all the macromolecules like proteins and lipids. They also contain carbon and hydrogen, right? So you can go through other reactions and produce this acetyl-CoA that can go and be used in the mitochondria to make ATP. So we can use other molecules as fuel, right? You can take fats and convert those into 2-carbon molecules and feed that into electron transport. You can do the same with proteins, but our body's primary fuel, its first fuel, is carbs. Then it can switch to lipids, and then, if need be, can use proteins for ATP production. All right, so let's summarize all this stuff. Let's put it together. So, for life to happen, again, life is not spontaneous, right? Or non-spontaneous. So you need energy to power the creation of these more complex systems. And that molecule that provides the energy, it's ATP. Now, where do we get ATP from? We get it from our food. We oxidize the food. Again, oxidation is a loss. So you strip away electrons and protons from food. You can use that to create gradients, like that chemiosmotic gradient in the intramembrane space of the mitochondrion. And then you can use that gradient to power the spinning of an enzyme, ATP synthase. And then ATP synthase makes ATP. So it's really crazy to me to think about how the energy for life comes from a spinning enzyme inside our mitochondria. And what makes that enzyme spin are the protons from the food we've eaten. But it's really spectacular. When we think about this, the flow of energy starts with the sun, right? Life only happens because of the sun, and life for us. But the energy from the sun is captured in photosynthetic organisms. We use that as food. Then we use that food and strip away protons and electrons from that and use that to make ATP. And then we can use ATP for cellular work, which gets released as heat out into the universe. So it's really pretty spectacular. Now, nutrients, on the other hand, nutrients get cycled. You're made of stuff that's billions of years old. You're just incorporating it into molecules of your body. So we're just constantly recycling stuff that's really, really old. So you're relatively young, but you're made of stuff that's billions of years old, and you're just combining it in new ways. So you can take pasta, eat your spaghetti, then you can turn that into CO2 that you breathe out. And then organisms of the earth can use that to make other molecules like potatoes. So it's really pretty amazing to think about the stuff that you consume, like a pretzel. This pretzel, I'll eat it, end up breathing it out, and then somebody else may use it as some spaghetti. So really pretty wild, but life finds a way. So what I'd encourage you to do at this time, summarize the big ideas in the lecture. There's a lot going on here, but if you get the big picture, start with the big picture, then you can make sense of things. I'd start with thinking about the words and how do you organize the words, like glycolysis, Krebs, oxidative phosphorylation, and then try to tie visuals to that, because a lot of us are visual learners. So if you can sort of bring that together and sort of visualize what's going on, you should be in really good shape. Now, if it doesn't all click today, no sweat. What we're going to do later is create one big document that puts it all together so you can see how it all works together. So if you don't have it today, just keep plugging away. You're going to get there. But thanks a lot, and choose wisely.